Hello friends, here I am having an exciting topic for you, that is biofortified maize, a best substitute to golden rice. As you know, the world is majorly affected with hidden hunger and protein energy malnutrition. Many of the African countries as well as Asian countries, the people are not having enough food to have. On the contrary, when people are having enough food also, there is no much nutrition. When we talked about Green Revolution, during that time, the major aim was to increase the food production, the quantity, but there was no quality which was targeted. So, that was the major cause for hidden hunger and protein energy malnutrition. During 2012, World Health Organization has summarized or they made a survey to see how much people are affected across the world, and according to them around 2 billion plus are affected with this particular kind of problem. So after that, many of the developing and developed countries of the world started to target about the enrichment of different food grains. For example, in rice, the targeted nutrients to deploy were zinc and iron. Similarly, in wheat, zinc and iron. In maize, beta carotene and zinc. In cassava, beta carotene. Beans, iron. Sweet potato, beta carotene. Pearl millet, iron and zinc. In banana and plantain, beta carotene. Here you need to remember is banana means banana which is consumed in ripe form, or else you can say when it gets ripe or yellow color or light brownish, we eat it as banana, and when banana is consumed in the form of vegetable, it is nothing but plantain. In both of these, the major aim or targeted nutrient was beta carotene. In lentil, a major pulse crop, here the targeted nutrient was iron. In potato, iron, and again in sorghum, it was iron. Biofortification. Fortification means enrichment of something, where biofortification means, it's a Greek word, bios means life, Latin word, fortification means making strong. Here you can see the inculcation of two words from two different languages. One is Greek, bios means life, and another is Latin word, fortification means make it strong. Biofortification is a process of generating genetically improved food crops that are rich in bioavailable micronutrients, either through conventional breeding or genetic modification. The main aim is make life strong. Why we need biofortification? Because as I told you, majority of our grains are deficient in vitamin C, B12, A, E and calcium. A cereal based diet is poor in specific micronutrient. Vegetables are rich in micronutrients. The presence of 21 nutrients in an average of 96 vegetables. Only B12, that is vitamin B12, is missing in case of vegetables. But the major thing is, even though vegetables are having enormous amount of nutrients, but because of the financial problems, everybody cannot afford it. So our major aim is to enrich the grain with these nutrients and to help the people over it. Implementing biofortification. For implementation, there is three broad questions which must be addressed. First one is, can breeding increase the micronutrient density in food staple to reach target levels that will have a measurable and significant impact on nutritional status? The second question is, when consumed under controlled conditions, will the extra nutrients bred into the food staples be bioavailable and absorbed at sufficient levels to improve micronutrient status? And the third question is, will farmers grow the biofortified varieties and will consumers buy and eat them in sufficient quantities? If these three questions are addressed, implementation of biofortification is very easy. Let us see the steps in implementation. So the first one is discovery, then we can say development, and finally dissemination. In case of discovery, we have three steps. That is, first one, identifying target population, then set nutrient target level, and the third one is screen germplasm and go for gene discovery, means identifying a particular kind of gene. The second step here you can find is the development. In development, breed biofortified crops. For this, test the performance of new crop varieties, measure nutrient retention in crop, and evaluate nutrient absorption and impact. The third step is dissemination. In this, develop strategies to disseminate the seed and promote marketing and consumption of biofortified crops. This whole leads to improving the nutritional status of target population. Let us see how it works. In case of traditional fortification, here we are having the common grains what we eat in day-to-day life. For this, we have to enrich with nutrients. In fortification, there are two things I would like to tell you, the difference between fortification and biofortification. You might have heard about the fortified salt. That means iodine is added to that particular salt externally. That means it is fortification. Likewise, you can fortify your crop when you supply with micronutrients either through fertigation or through by soil application. Okay, that is nothing but fortification. But when it comes to biofortification, that means you are just giving the nutrients, or you are fortifying something internally so that it will give a long lasting effect. That means heritable effect can be found in that particular individual. So in traditional fortification, we are taking the normal grain, then we are taking nutrients by food processing, cereal with added nutrient, then dietary supplements are added, and because of all this process, more nutrients are consumed here. Okay, this is the first step, or we can say the traditional fortification. When it comes to biofortification, here selective breeding strategy is followed. Selective breeding strategy, simple breeding techniques, back crossing, pedigree method, all these things can be followed in biofortification. First, what we'll do in the selective breeding, we take the plants producing low level of nutrients, then breed the plants and select those with high nutrient level, means we will go for selection. For this, we will go for genetic modification, or by just selecting already available best plants or best lines with fortified or already enriched, or the plants or lines which are already having the good nutritional quality are selected. The other option is going for genetic modification. From this pool, we can select the plants or lines with higher nutrient level, and altogether we can go for plant producing high level of nutrients. This will be giving more nutrients for human consumption. Now the picture was very simple and talking is very easy, but let us see how fortification is achieved. It may be achieved through conventional molecular, transgenic or RNA interference approaches. Application of micronutrient either through soil or foliar spray. This is nothing but the fortification. Okay, Then transgenic approaches, this is mainly used for biofortification. Then conventional breeding approaches like through backcrossing and pedigree, as I mentioned already. Then marker assisted breeding, this became more popular these days, where gene stacking or gene pyramiding is done in order to incorporate two or more micronutrients into a single line. Then marker assisted backcrossing for identifying QTLs, controlling these nutrients availability and transferring to the desired plants. QTLs are used when a trait is governed by quantitative gene or quantitative trait, and that is controlled by minor genes. In that case, marker assisted backcrossing is a successful approach. So transgenic approach is one more, where the genes which are available either in plants or in animals or anything, wherever it is available, they have just chosen it and introduced into a particular plant to enrich or to increase the nutrient level. Let's see the advantages of biofortification. It capitalizes the regular daily intake of food staples, implicity or targets low income households. After the one-time investment to develop fortified seeds, recurrent costs are low, and fortified seeds shared internationally, which is cost effective. Fortified seed does not incur a yield penalty that may have important indirect effect in increasing farm productivity by helping plants resist disease and other environmental stressors. Next, we'll talk about the golden rice project. Unless we talk about the transgenic approach, we won't get to know the importance of biofortification. As you know, golden rice project started during 1982 when Ford Foundation started helping for this particular project. Ingo Potrakis was the major scientist who did his hard work in this particular aspect. Here in wild type, green or golden rice 1 and 2 are the two developmental approaches, where golden rice 1 was having 1.6 micrograms per gram of beta carotene content, and golden rice 2 was having around 32 to 36 micrograms per gram of the grain. In golden rice 1, there were three major genes. PSY, which was taken from phytoenzyme, source was daffodil. Similarly, ICY was where enzyme was lycopene beta cyclase, and this was also taken from daffodil. And one more gene was CTRL, that enzyme encoding was phyto-N desaturase, that was taken from bacteria Erwinia iridovora. But when golden rice came to the market, the major problem was the amount of beta carotene what was available in that was not sufficient for in the form of whatever the rice consumed per day. For that, scientists again started an approach in order to increase the content of beta carotene, and finally they got a source, that was maize. As golden rice 2 contains the bacterial CRT1 and PSY, that is from maize, just like the previous version, but genes are under endosperm specific promoter control. The selectable marker agent is mannose. Increase in provitamin A content is about 10-fold in this one GR1 or golden rice 1, and about 25-fold increase in prototype. Golden rice is mainly breeding project today, like Philippines, Vietnam, India, Bangladesh. Introgressing 8 events into 11 varieties was a major thing. Today, golden rice cultivation is not adopted in India because, as you know, the transgenic approach is uh, discouraged in India after the failure of, we can say, the BT cotton, but many of the countries across the world are using this golden rice and they are cultivating, like from since 2018 Canada, Australia, New Zealand and USA they approved golden rice cultivation. Since February 2019, Bangladesh Agriculture Minister Abdul Razzaq announced that they will start cultivating golden rice in future days. The difference between golden rice 1 and 2: 35 micrograms of carotenoid per gram of dry golden rice 2 instead of 1.6 microgram of carotenoid per gram of dry golden rice 1. More efficient 5 gene introduced in golden rice 2, but the limitations are acceptability and ethical issues, superweed development, lacking other nutrients, and fear of losing indigenous cultivars, as we are having fear about other transgenic crops. Coming to the factors considered to breed for micronutrient biofortification, first one is retention and bioavailability of target nutrients, then breeding techniques to be employed, nutritional factors and promoters, post-harvest losses. Let us see the difference between fortified maize and golden rice. The breeding procedure in fortified maize is conventional, whereas in golden rice it is genetic engineering. Fortified maize is fortified with many micro and essential mineral nutrients, whereas golden rice is enriched with beta carotene only. In fortified maize, the seed availability in market is easy, means easily we can buy it, but golden rice, when genetic engineering is used, seed may be costly and less available. Effect on yield of fortified maize is moderately effect on overall yield. On golden rice, no much effect has found. Acceptability of fortified maize is highly accepted around the world, but golden rice accepted mainly only in the developed countries, that too very few. Coming to maize, or we can say what the major discussion we are doing here is why maize. As you know, quality protein maize is already a popular aspect. Baby corn, sweet corn, popcorn, single cross hybrids are available already. Well developed inbred lines are available. Technology for single cross hybrid seed production and commercial cultivation for normal QPM and specialty corn are also available. Vitamin A fortification in India, that is nothing but beta carotene. Allied maize parents used in this approach are VQL1, VQL2, V335, V345, HKI1105, HKI323 and HKI161 through marker assisted selection. Transgenic approach is also used here, but it is of not much importance, because when golden rice was developed, the genes were taken from our maize only. The main thing is why the people won't consider maize as a staple food, because from the ancient periods it was continuously fed to the cattle and poultry, but nowadays when non-vegetarian numbers are increasing, as well as when people are also adopting to maize as their staple food, biofortification in maize is a great achievement. However, transgenic approach is also used here, like CRTB, that is phytosynthesis, and CRT1, that is keratin desaturase. Genes from Erwinia herbicola under the control of G gene promoter resulted in accumulation of 10 micrograms per gram of beta carotene in maize genotypes. Transformed white maize genotypes, that is EM37W, with combination of 5 genes, that is PSI1, CRT1, LYCB, BCH and CRTW. As you remember, in rice there were only 3 genes. 2 were taken from daffodil, again they were replaced with maize, and 1 was from Erwinia eurodobora. Okay, but here 5 genes. Bioavailability of nutrients and low phytate content. In maize, high phytate reduces the availability of iron and zinc. As you know, the nutrient status are interrelated. If you want to increase one, automatically the another one will decrease. Likewise, when we don't need the negative factor, by decreasing it we can increase the positive factors which we want. Low phytate deteriorates seed quality. Optimum is favorable. Various low phytic acids like LPA1, LPA2, LPA241 and LPA3 mutations have been isolated in maize. To introgress these mutants, marker-assisted bacterospreading is used. Low phytate QPM, good for human and meat industry. Coming to QPM, the content of normal maize, and we will see the content of biofortified maize. Content of normal maize is 1.3 grams per 100 gram of endosperm protein, whereas it is 55% more tryptophan, 30% more lysine and 38% less leucine than that of normal maize in case of fortified maize. In normal phase, the protein content is 45%, whereas in fortified QPM it is 80%. In normal maize, the protein intake utilization is 37%, and in fortified QPM it is around 74%. Nitrogen equilibrium is 24 gram in normal maize, and it is around 8 gram in case of fortified QPM. Coming to the traits considered to develop the fortified maize, the first one is genetic variation with desired agronomic character. For biofortification, the basic requirement is genetic variation. As you know, this is one of the basic principles of breeding, and with the desired agronomic characters. Then consumer acceptance. If you have developed any variety with very nice traits, just like the initial wheat varieties in India, if it happens, if a good variety also, if consumer preference is not there, it is a big failure. When consumer acceptance means where yellow grained maize is not very popular. As you know, in India, white flour is much preferred over any other color of the flour. Likewise, if yellowish stain is present in the flour, nobody will prefer. Likewise, if white maize is there, it will be preferred by majority of the people. Nutrient losses during different stages of crop handling, the bioconversion efficiency and bioavailability of nutrients. Finally, health, technology dissemination and seed systems of the country. The institutes involved in biofortification of maize, first and foremost, is Consultative Group on International Agriculture Research through Harvest Plus program, then Indian Council of Agriculture Research, Department of Biotechnology, National Agriculture Research System, or in short we call it as NARS, then BISA, that is Borlaug Institute for Southern Asia, and many other agriculture universities are actively participating in this fortification approach. Coming to popularizing biofortified crops, QPMs like Pusa Shakti, Kheer Mix, Dil Khush, Khadi Mix, Protino H are sold, like QPMs are sold in all these names, like I'll repeat it: Pusa Shakti, then Kheer Mix, Dil Khush, Khadi Mix, Protino H, with different names. It is sold in Rajasthan state. Then educating rural masses by gram panchayats and sensitization of health centers on the health creates opportunities to develop small scale industry and empower village women. Expansion of value addition industry would enhance the adoption. Then adoption means it improves the diversification in crop. As you know, India is basically dependent on rice, wheat. Then if we leave these two major cereals, we may be adapted to ragi, sorghum, like that, but not to the minor millets or maize. If we go for such kind of approaches like biofortification, adoption will be greatly promoted. Okay, the effects of policymakers would definitely help in popularizing biofortified maize in India. Like good policy makers or good policies must be and emphasized through government so that it can be popularized. Thank you. I hope you liked this video. If you liked, please do not forget to hit the subscribe or hit the like button. Thank you.